This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1045, recorded on September 15, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hi, everybody. I'm just booting up the weather here. Yes, Austin, Texas. Get this, 84 degrees. Man, that's like 20 degrees less than when I last spoke to you. And there's been rain the past couple of days, and so we're all really happy about that. It's I guess it's clearing up now, so it's sunny-ish, but I think we're out of the worst of it.
It's gotten much cooler here. I can't find the temp. I'll let Brienne do it. Also joining us from Madison, New Jersey, Brienne Barker.
Hi, it's great to be here. And here it is 72 Fahrenheit and beautifully sunny. We've had a lovely last couple of days.
From Western Massachusetts, Alan Dove.
Good to be here. It is 67 Fahrenheit, 19°C. Overcast, and we have somewhere far out off the Cape, somewhere to our east, is remnants of Tropical Storm Lee, which is supposed to bring storm surge to the Cape and Islands. And we're getting no rain out of it, which is a nice relief because we have been deluged with rain for the past couple of weeks.
And from Montreal, Canada, Angela Mingorelli.
Hey, everyone. So for the first time, maybe the first time, it's not the coldest in Montreal. So it's 70 Fahrenheit and 21 C. So it's a bit colder where Alan's at. And it's partially cloudy, kind of sunny-ish. I'm not complaining. We've also had a lot of rain and it's been getting pretty cold at night. You can definitely feel the fall in the air.
Yeah, all of a sudden it got chilly. Oh, well, it is September 15th after all.
We had a heat wave last week, though. It was really hot here. I'm not sure if everyone else also experienced this. Or maybe was it 10 days ago now? Maybe like almost 2 weeks ago. It was really hot.
What's really hot?
Yeah,
like
we had
36 C? What's that? Like 37?
Yeah, that's like 90 Fahrenheit. 95.
For Rich, it's not that hot, but for us.
No, for Rich, that's just like ordinary spring morning. But
Yeah, I think I remember that.
Mm-hmm.
My weather disappeared from the front of the phone. I don't know why. New York is 22 C, and it's cloudy here in New York. It's very different from you, Brienne. There's no sun here.
Oh,
It's very interesting.
there's still sun here.
Yeah. All right, we have a couple of announcements. First of all, if you like our work, we'd love your support, your financial support. It doesn't have to be huge, but it needs to be. It has to exist. And you can go to microbe.tv/contribute to do that. You know, as the pandemic recedes, people forget about viruses, but we'd like to keep on communicating. Microbe TV does more than viruses, does many other cool things, and please help us do that. Microbe.tv/contribute. I want to wish Fiona a happy 40th birthday.
happy birthday, Fiona.
Happy birthday, Fiona.
It was September 7th, and hope you had a good time. Fiona is a PhD in psychology and a massive TWiV fan.
Cool.
And, her
Happy
husband,
birthday.
her husband,
First anniversary of your 39th birthday is always a good time.
I think you could probably do a PhD in psychology shrinking the TWiV hosts.
oh boy, indeed. You don't want to go into our heads. I don't think it's a good thing.
No.
You don't want to be there.
But her husband, who wrote in saying, would you please do this, said she probably should be a virologist. She loves TWiV. So thanks for listening. Keep it up, Fiona. Many more. Don't forget, there is a second position in Amy Rosenfeld's lab at the FDA. This is a research assistant to work on enteroviruses, making animal models, understanding the consequence of cross-reactive antibody responses, doing cell culture, all BSL-2 work. We'll have a link in the show notes, and you can find an email to find out more. And sadly, I want to tell everyone that Robert Lamb, or Bob Lamb as we used to call him, passed away on September 2nd. He was a very important influenza virologist who was on TWiV a couple of times and who was always very good science, but also a very enjoyable person. So we will miss him. Sorry to lose him so early.
Yeah.
He was a postdoc with Pernell Chopin at Rockefeller University, which is where I first met him. He was on my qualifying exam committee, and he had a wonderful British accent, and he was quick and funny and a lot of fun. I always sought him out at meetings because it was fun to talk to him. And then for most of his career, he was at Northwestern University. So sorry to see you go, Bob.
Nice picture of Bob here on the obituary that you found.
I hope he finds some good plaque assays wherever he goes.
Plaque assay heaven.
Plaque assay heaven. Yeah, that's a good one. All right, we have two very cool papers for you today. The first, our snippet, is in Nature Communications. A Lassa virus mRNA vaccine confers protection, but does not require neutralizing antibody in a guinea pig model of infection. And that's why I want you to know about this paper. You don't need antibody, apparently. We'll go through it, but very interesting.
Well, you don't need neutralizing antibody.
You don't need neutralizing
antibody, yes.
Yes, that's a title, neutralizing antibody. And so this is two first authors, Adam Ronk and Nicole Lloyd, and two last authors, Andrea Carfi and Alexander Bukrayev, who I noticed on LinkedIn. He just was talking about this paper, and I said, we're doing it on TWiV right now. This is from University of Texas, Galveston, Harvard, Scripps, La Jolla Institute for Immunology, and the Academic Medical Center in the Netherlands, and Moderna. So there are a couple of vaccines in development for Lassa fever. This is one of them, but the antibody, the neutralizing antibody angle is very interesting. Lassa virus is the virus that got me interested in virology. Honestly, it really is. I read this book, Fever, after I graduated from college, and I said, oh my gosh, viruses can go from animals to humans. And I said, that's a cool thing. I'm trying to rearrange my monitor here. That's why I'm looking funny. There we go. That's a cool thing. And so based on this book by John Fuller, which is very cool, you should read it. I looked into getting into virology. So Lassa virus, it's an enveloped RNA virus, negative stranded. It causes up between 100 and 300,000 cases every year, primarily in Africa. First found in Lassa, Nigeria, about 5,000 deaths. And, this is a medical need for sure. 5,000 deaths, 300,000 cases. They often go to other countries. So
and that's 5,000. That's 5,000 deaths a year, right? Not in total.
Yeah.
That's yearly.
Yeah.
Exactly.
Mhm.
I just wanted to clarify.
That we know about. That
we know of.
That
Exactly.
we're diagnosed with this virus.
No, I mean just for the
which-
listeners, because
Yeah,
Vincent didn't
that's
say
5,000 a
year.
year. Yes,
Yeah.
not since the beginning of
Yes,
time.
a year.
Which is a lot.
And this is a virus that is it is from animals. It comes from rodents into humans through rodent waste. So it's in conditions where people are around peridomestic rodents, and there are a couple of species that are especially good at carrying it. And they basically inhale the fumes and get the virus.
Most of the
I was impressed here. It mentioned something about how long some of the virus could be seen in post-exposure, at least in human urine and semen. And I was like, whoa.
Yeah.
There's a lot of potential reservoir for this beyond what I had realized.
So I've done a couple of Lassa episodes with experts, and they say most of the transmission is actually spillover from rodents. There's some human, but not a lot. Most of it is a rodent spillover. You live in a house where there are rodents under the floor, and that's how you get it, which is amazing, right?
Is the geographical distribution constrained primarily by the distribution of the host, the rodent?
The rodent host, yeah.
I suspect so.
And there are more.
And the other thing that I was unaware of, or if I knew, had forgotten, is that it says that upwards of 80% of the cases are asymptomatic.
Yep.
Yeah,
So
I didn't know that
it
either.
may be underreported, right? Yeah.
Yeah, so they've got to figure that 100,000 to 300,000 human cases, that's known human cases. Probably a lot more people are infected because it turns out you can get this and not even know.
Yeah.
It's a virus you need to work with under BSL-4 conditions.
Yes.
And that's the Galveston contribution here, the BSL-4.
And that's an important thing to remember going through this paper, because as I was reading, I would occasionally say, "Oh, well, why didn't they?" And then, "Oh, right, yeah, they're in bubble suits is why they didn't." You can't just blast through your experiments like this.
Yes,
This,
very
is,
expensive. This was
very-
all done in a
very
BSL-4.
expensive and-
and difficult.
and very time consuming, because
Yeah.
everything you do, you gotta suit up and go in, and
This is one of the viruses on the WHO blueprint for the diseases that need accelerated research, right? And so this one is definitely one where we need a vaccine. There will be a vaccine at some point used, maybe multiple, but this is just one of them. And it's, of course, this mRNA vaccine it was done because of the success with the SARS-CoV-2 vaccines. And so they have taken the mRNA encoding the spike glycoprotein of the virus. They have made two versions, one a prefusion version, very much like the spike prefusion, to lock it in a version that displays the relevant antigenic sites. And then they also use one without the prefusion locking to compare it. They put it in lipid nanoparticles, and they deliver it to guinea pigs, which are the model for Lassa disease. These are outbred animals. Okay, so remember that there's not a lot of immunological agents reagents available, and so forth.
and I'm assuming somebody's tried this in mice and it just doesn't work, because it's
so when
mice.
good
There
question.
are mouse species that are normally infected with this going around spreading
So
into humans.
when I was at the Rocky Mountain Labs, they were trying to establish a colony of Mastomus natalentis, which is the natural reservoir. So that's not easy. You have to catch them, and then you bring them, and you get them to breed. And he said it's hard to breed them in captivity. So this was a couple of years ago, but I think that's the need. Mus musculus just doesn't work, right?
And I imagine there would be containment concerns with bringing another wild mouse into the country and breeding up a bunch of it when that mouse is endemic to many parts of Africa and is a reservoir for this.
Definitely.
Yeah, I mean, that would be a problem. And then if you wanted your model to have disease, particularly because they mentioned this is lethal challenge, if some of these other rodents are reservoirs, they may have completely asymptomatic infection. And unless you're going to ask them about going to the opera, you are not going to be able to have a lot of data on protection in your challenge model.
Yeah.
So we use guinea pigs, and they are vaccinated intramuscularly with these lipid nanoparticles containing the spike mRNA, which has been pseudo-uridylated, so it chemically modified, so it doesn't induce responses that would interfere with it. The controls are phosphate buffered saline infected guinea pigs, and then they look for first binding antibodies, so antibodies that will just bind the spike, and they do find those. And in fact, both constructs induce binding antibodies. And then they look for neutralizing antibodies. So they do a neutralization assay. And some of the sera, with the wild type mRNA encoding the wild type spike without prefusion stabilization, none of the sera demonstrate neutralizing activity over the background found in the pre-immune serum. So basically no neutralization. And in the pre-fusion stabilized vaccinated group, 2 out of 5 animals have neutralizing activity. Right. So 2 out of 5 animals have
And even then,
neutralizing.
it's not particularly impressively high.
Yeah. And they say this variation from animal to animal is not surprising because they're outbred animals, right? And so they're genetically diverse.
And that's, they assay neutralization. Maybe you said this. They assay neutralization on virus,
Virus. Yeah.
right? And they have a plaque assay.
Let's see. Let's go to the methods here.
I didn't look closely enough at that.
I'm gonna do a thing which is plaque. Let's search for plaque and see if
That's what I was
plaques
doing.
were visualized.
Yeah.
Yeah, they did a plaque
Yeah.
assay.
All right.
Look at that.
Good for them.
Yeah, good for them.
So that's all BSL-4 work.
Yeah.
Yeah, so the guinea pigs can be immunized outside the BSL-4, and then when you want to challenge them, which we haven't done yet, we bring them inside the BSL-4, obviously.
So it's relevant to point out that one of the things that they point out is that the neutralizing antibodies that they know of that have been identified in the past are all 2 quaternary epitopes, meaning they're conformational epitopes that are not apparent unless you have the whole trimer assembled,
that's right.
Brienne?
Correct.
So that means that they're pretty complex epitopes.
They bridge like 2 subunits of a trimer, right?
Yeah, and so maybe it's not, although I expect with an mRNA vaccine that this protein will assemble in a membrane in a fashion that mimics its assembly on the virus. Maybe it's not quite like that, or maybe something about the presentation fails to present the appropriate quaternary epitope, or maybe there's something else about an infection that induces those antibodies. Do you have any thoughts about that, Brienne?
I don't know enough about Lassa viral particles to really say much about that, to know if, say, there are other proteins that may assist in that quaternary structure, perhaps, or something like that.
This is the only membrane protein in the virus.
Okay.
There's nothing else as far as we know, yeah.
I suppose I assume there's a matrix protein of some sort.
Yeah, there
I
is,
suppose
yeah.
that could play a role, at least in
Right.
organizing the glycoprotein on the surface.
Yeah.
Yeah, I just don't know enough about that to really tell you, but I
Right.
agree with everything that you're saying about what that quaternary epitope means.
They do some experiments to map the epitopes recognized by these antibodies, but I'm going to skip those because I don't think they're part of what I'm interested in here. If that's okay with everyone, does everyone?
I'm cool.
You're
That is
cool with
totally
Totally
fine.
Okay.
Totally fine.
Good. Now, this is very interesting. The next section in the paper, they say, despite excellent protection observed in each vaccinated animal, we decided to look at the mechanisms of protection, but they haven't
They
presented.
haven't checked for protection yet.
Why did they? Did you think it was a mistake, or it's weird that they would present this before the protection experiments, right?
Yeah, I think that's just an issue with drafting the paper, that they phrased that poorly, because at this point in the paper, if you are following in sequence, and as we mentioned a number of times on TWiV, when you read a paper, the sequence in which the experiments are presented is seldom the sequence in which the experiments were done. So, they obviously knew when they were writing the paper what the outcome was.
Yeah,
but at that point, if you're reading it, it's like,
Funny.
wait,
I would have
excellent
written
protection.
I would have next gone to the protection and
Yeah.
then
Yeah.
Me too.
put the next one. So I'm going to go right now to the protection because I think
Yeah.
it makes more sense. Okay. So
Freestyle.
56. Yeah, we can. In the old days, you could cut up the paper and repaste it. Day 56 after immunization, they infect the animals intraperitoneally with 30,000 PFU of a guinea pig adapted Lassa virus, Hosea strain. I like the name, Hosea.
Yeah.
And they record weight, temperature, disease scores every day. Control animals, that is BBS, no lethargy, ruffled fur, weight loss, or other signs until 7 to 9 days after challenge, and then they became febrile. They began to lose weight. They got lethargic, ruffled fur, orbital tightening. Their Earth around the sun
They
orbit got
went into
tighter.
smaller and smaller. They're coming down into the atmosphere.
Well, that's part of like the grimace, right? So if they're in pain,
Yeah.
then they'll.
Yeah,
For the listeners, that's a
Oh yeah,
Yes.
cool.
yeah. It's a part
Well,
of the
not cool,
grimace
but thank you
score.
for the explanation.
Yeah.
They became viremic by day 9, and they met the criteria for euthanasia. So they were euthanized on days 11 to 15. Vaccinated animals, both the wild-type glycoprotein mRNA and the prefusion stabilized, no outward signs of disease. They all survived challenge to day 28 when they were euthanized to take a look at the tissues. So, none of these signs, no ruffled fur, no orbital tightening, none of that. They had an increase in glycoprotein antibodies, a little increase in nucleoprotein antibodies, and they say they never find virus in serum after challenge, which is very interesting. But they say it must have been a little replication because there's antibodies to nucleoprotein.
But it's surprising that the clinical score is the exact same. Like there's none of the animals that were vaccinated. Like there's no variation whatsoever, or even like you were saying, the viremia is under the limit of detection. Like
So
yeah, it looks like the only thing that maybe changes a little bit is like they get a teensy bit of temperature.
which is interesting,
Right.
actually. The only it's more than you would expect in the wild type GPC vaccine. Like in the I'm looking at the figure, but definitely not almost the same as the control, but similar to yeah.
They do histology, and the main sites of disease in the control group are the lungs and the liver. You see typical viral pneumonia, interstitial pneumonia, and sections of liver showing pathology typical of Lassa virus infection. And the vaccinated animals with either vaccine, normal lung, normal liver. So both of these mRNA vaccines protect against death and severe disease, clearly. All right. And so now we can go back one section and say, what is protecting these animals? So they say, despite excellent protection, there's not neutralizing antibody in every hamster. And as Brianne said, even in the ones that do have it, it's not great. So what are they thinking? Well, they're thinking about other mechanisms involving antibodies, and particularly the FC portion. And Brianne has a show and tell for us.
I do. I talked about these exact functions today in class, and so I already had this out on my desk so that we can see an FC portion on an antibody.
Yep.
So that's below
and
the
for those
eyes,
of you who are
right?
listening, it looks like a very cute stuffed lobster.
I was gonna say the same thing. It looks like a
Yeah, it's
lobster.
So the FC portion combined receptors on a variety of cells, and those cells can then mediate clearance of virus by the antibody recognizing the antigen, say, on a cell surface. And so they tested antibody-dependent neutrophil phagocytosis, antibody-dependent cellular phagocytosis mediated by monocytes, antibody-dependent NK cell activation, and antibody-dependent complement deposition. So the antibody can also activate the complement system. And so I guess, Brianne, these are all acting on infected cells, or some would also virus particles, maybe?
So it can be virus particles or infected cells. The NK activation is definitely acting on infected cells.
Yeah.
I usually think of the phagocytosis and the complement deposition as acting more on viral particles. But you could perhaps come up with ways that it could be else.
So wait a minute. Let me make sure I got this straight. If you say acting, first of all, this has to do with the claw part of the lobster antibody binding to antigen.
Yes.
Okay.
Viral
and then
antigen,
the tail part of the antibody, which is the FC portion, binding to something else and affecting the fate of that antibody antigen complex, right?
Correct.
So when you say acting on infected cells. Do you mean the antibody is binding to protein on the surface of infected cells?
Yes. And so
So,
with an enveloped virus, you would see some amount of protein in the plasma membrane, and antibodies could bind that protein.
let's go through again. You said some would be acting on cells, other antibodies.
So that would be an antibody-dependent NK activation. So you would have some protein on the surface of the infected cell, antibodies would bind, and then NK cells would kill the infected cell.
Okay. And then there are a couple of phagocytosis assays. That's where there's some sort of FC receptor on the cell that
On the phagocyte.
on the phagocyte that just sucks up the whole antibody antigen complex and gets rid of it, right?
So it
In which
basically
case it's
makes
bound to
it-
virus, right?
Yeah, so it's probably virus, and it's probably enhancing that virus's phagocytosis.
And the complement, is that the complement going to be acting on the virus?
That's tougher, but I think the complement would probably be acting on the virus. I don't know if the pore, it would be membrane attack and making a pore. You can also think about complement helping phagocytosis, or complement inducing some inflammatory response. I don't know enough about their ADCD assay that they use to know exactly how it's being measured. I think they're just looking for complement being deposited. They don't actually look at what the deposited complement does.
But importantly, these are all ways to get rid of either virus-infected cells or virus that don't involve neutralization.
Absolutely.
Yeah, exactly.
And the
And they all require the FC portion,
Okay.
Right.
which neutralization might not.
And they find evidence for all of them in their assays. They have a variety of assays to measure these. And they say vaccination with either construct induced each of these FC-mediated immune mechanisms. So if you wanted to take this further, Brianne, what would you do to show that these are working in the animal, say? You could do an NK null guinea pig if you could do that, right?
You can do an NK null guinea pig. You could... I would be interested in knowing exactly which types of antibodies they are, because different antibodies, say IgG versus IgM, tend to do some of these functions more or less than others. So IgM, for example, is very good at fixing complement. And so if I saw that the animals were largely making M, then I could start to look at what's going on with complement a little more.
So does this mean that you would get similar results in humans?
I think you'd have to check and see.
Yes.
Actually, they say they're going to do non-human primates next, because that this doesn't mean anything about people. I would also say that non-human primates not going to definitely tell you, but it's the best you can do. And then you go into, you do a phase 1 and phase 2 and phase 3. You certainly have enough cases to do phase 2 and 3, right?
Right.
If you
Mice
can get
lie
to them.
and monkeys exaggerate, and I'm not sure about guinea pigs.
Yeah.
Guinea pigs laugh. So that's the interesting part of this. I think it's quite interesting that this vaccine is protective. Looks like infection because they don't see viremia without neutralizing antibody. And the conventional wisdom is you need neutralizing antibodies to prevent infection. But here's an example, and it may be that the replication cycle is sufficiently long of this virus that it gives time for FC-dependent mechanisms to kick in. I don't know. But I thought that was really interesting.
It is. They also do mention that they have not looked at cell-mediated immune responses here, so we don't know if T cells are doing anything
That's a good
in
point.
that protection. So that could perhaps be another study as well.
Yeah, which that's something that's limited apparently by the available tools for guinea pigs.
Right.
So they just don't have assays that they can use there.
Brianne.
Yes.
If you vaccinate with this mRNA vaccine, how many days later will you get effector T cells at the sites of injection? What are they challenging with them? Intraperitoneally is the challenge?
They're challenging intraperitoneally.
So how long would it take to get T cells there?
Between a week and 2 weeks.
So
They aren't challenging till 56 days.
Yeah.
Yeah.
Because it
Yeah, but
depends
this-
on
Yeah, and they
Lassa,
gave-
right? Because certain viruses, it's very well defined. Like LCMV, you know that like by day 8, you'll have like peak T cell response, and then you'll be able to.
Right.
But with Lassa,
Right.
has that
Yeah,
even been
maybe it's
studied?
different.
I have no idea.
I don't know.
Yeah.
That's a good point. But you know that remember that in the control animals, they have no signs of disease until 7 to 9 days. So presumably the virus is reproducing, I mean, maybe that's when the T cells would be coming up, depending on the kinetics, right?
Absolutely.
But anyway,
And we talk about that with, day 8 or whatever as being kind of the peak response. Earlier, you'd see some response, just not
Yeah.
the best possible response.
anyway.
So when a new variant of SARS-CoV-2 shows up,
Mhm.
and they look to see whether that variant is how it reacts with serum from previous infections.
Right.
The gold standard, as it were, they're usually reporting on a neutralization assay.
That's right.
And if they say the serum that was from previous infections does not neutralize or poorly neutralizes this new variant, you don't necessarily have to freak out. Because
Depends
there's a lot of other stuff going on.
Depends how old you are and how many comorbidities you have, right?
Right.
Because there you might want to freak out. But yeah, a lot of people who have only the 3 original SARS-CoV-2 vaccines who then get infected with Omicrons, they stay out of the hospital, right? They're alive because of probably some of these effector functions and T cells and other, I guess. But still,
of course, with all the caveats involved. This is guinea pigs,
Yes.
it's Lassa, it's not SARS, blah, blah, blah.
no, I mean, I think most people will say that that antibodies are really important for preventing infection by SARS-CoV-2. But if they don't neutralize, you can still live. And some studies have shown, and we've done them, that FC-mediated mechanisms are involved in protection in an animal model. We did that. I think that was a Michael Diamond paper. Sometime
Yeah, for some reason I was thinking we talked about it or somehow referenced Paul B. Nash and Theodora
maybe
during that discussion.
So I think, Anyway, there are a number of other vaccines in development. I just want you to know that they're 2 VSV, vesicular stomatized vectored vaccines, and there are they're actually DNA vaccines as well. So there are a bunch of others, which is good because we want to have a vaccine and vaccine. We don't need to have 5,000 deaths a year from this infection.
Yeah, I was impressed they summarized in the paper the different candidates that are out there, and I didn't realize there were that many.
Yeah.
Right.
For this virus. It is a very serious ongoing development effort. And it seems likely that something's going to work.
They also pointed out that there were differences in the immune response to those different vaccines. Was that I forget, and I'm trying to find it. Was
there
that
were some
whether or not they induced these quaternary epitope neutralizing antibodies?
Yes. Yeah, there were some that seemed to work. There were some others, I think, that seemed to work even though they don't always induce neutralizing antibody or moderate neutralizing antibody. So this may be a theme.
So there is
looking at, the sort of the composition of those different vaccines, I don't see any way you could predict that they would give different sorts of immunizations. A lot of them are making protein during the immunization. Okay, so
yeah, one's an attenuated virus. A couple of them, I think, are vectored.
Yep.
So there are a couple of measles virus vectored constructs that don't induce neutralizing antibodies, and they still give near sterilizing protection, they say, in non-human primates. So this is something that we're seeing with Lassa virus, I suppose. So anyway, that's that. I thought that was cool.
Very cool. I like that. Thank you.
Yeah.
Next paper is, this paper makes me obsolete. Just kidding. This is an
Yeah, but they cited you, okay? So
I'm very impressed. They cited me and Charlie Weissman, and you know, most people forget what happens more than 5 years ago.
that's right.
Science.
Well, I missed that. What'd they cite you for?
Oh, for
Okay, so in the discussion,
yeah.
here, this is the sentence, and this is a good way to start this, actually. Several years after the Asilomar Conference on Recombinant DNA in 1975, the first complementary DNA clones of RNA viruses were created.
Okay.
References 25 and 26, Charles Weissman and Baltimore and Racaniello. So
Cool.
when I went to David's lab in 1979, he said, Vinny, Asilomar said we could clone the whole polio genome. I want you to do that and see if it's infectious. And I did it, and it was infectious, and that's where this came from, right? You can make a DNA copy of poliovirus, RNA genome, so now you could manipulate the genome because you couldn't manipulate RNA.
Yeah.
And now this paper is going to change that. It's going to...
And that has been, we've talked about this off and on various episodes. That's been a huge roadblock to studying RNA viruses in general because, I mean, Vincent did this with poliovirus. It turns out you can't just do the same thing with every other RNA virus. Some of them you can, some of them you can't. You get the cDNA. That part is now relatively straightforward, but whether or not it's infectious depends a lot virus to virus. So there are a lot of RNA viruses that we can't do molecular biology on very easily, or at least we couldn't. And that's what these folks are trying to address.
Yeah. And I mean, you could imagine other ways that somebody might think about wanting to edit RNA, and editing RNA basically wasn't a choice.
Yeah.
I mean, they mentioned, SARS-CoV-2, you couldn't take a single 30,000 base piece of DNA and stick it in a plasmid. You had to make fragments and clone them in multiple plasmids and put them in cells so they recombine and so forth. So it was tricky. But in theory, this method, which is in this paper, could get around that. It's called CRISPR-based engineering of RNA viruses by Artem Nemudri, Anna Nemudraya, Joseph Nichols, Andrew Scherfius, Trevor Zoll, and Blake Wiedenheft. And they're from Montana State University in Bozeman, Montana. So I'm thinking, okay, where is this? UCSF, Chicago, Stanford, Harvard? No, it's Bozeman. So good work can be done anywhere. Just need you need a lot of things. You need money, you need interest, and so forth. Anyway,
But it's only 6 authors on this paper.
No,
Yeah.
it's cool.
Yeah.
It's very cool.
Not an enormous group these days.
Hey, when I did this with David, there were 2 authors on the paper.
Yeah, well, the different times.
Those are the old days when we walked uphill 30 miles to get to school in the snow.
Both ways, yeah.
Anyway, you can... They make this very interesting historical statement. Paul Berg was the first to make a recombinant DNA molecule in 1972. And then we have used recombinant DNA to study many, many things, not just viruses. And then CRISPR came along to allow editing of DNAs, but very few tools to do RNA. So just to remind you, CRISPR is a defense system from bacteria and archaea that evolved to protect against viruses and incoming DNAs into bacteria and archaea. And it involves a system where a set of nucleases in the host cell combined with a guide RNA that targets a specific sequence will say if a phage infects a cell, the DNA will be degraded. And it's been repurposed by scientists to do gene editing. So you can take a guide RNA and the proteins, the nucleases and so forth, and put them in cells, and you can edit DNA in reasonably precise ways. But we haven't been able to do that with RNA. And that's what they're going to do in this paper.
Yeah. I really liked how they laid this out in the introduction.
Yeah.
Because they kind of they made it in a way that I'm interested to think about with students, because they basically talk about the fact that, well, if you're going to manipulate DNA, you need an enzyme that cuts the DNA in a place where you want, or some way to get the DNA cut where you want to, and you need repair. And fortunately, our cells can do lots of DNA repair. So we just cut the DNA where we want, and then we set up repair in a useful way. And they talk a little bit about the fact that, well, first we need to have some way to cut RNA, and we found some of that with other CRISPRs, but they don't always cut in exactly the right place. And as soon as I started reading this, that approach of, well, first you need to cut, then you need to repair, just made it very clear in my mind of like, oh yeah, RNA repair.
Yeah.
What? That's
Nobody
you can't do that.
repairs
How are they
RNA.
gonna,
It's
how are we going to
disposable.
repair RNA?
And the CRISPR system evolved as a way to destroy RNA.
Sure.
So you're not going to find much luck there, right?
Mm-hmm.
So there are many different kinds of DNA CRISPR systems, and there are also some that target RNA, and the one they're going to use here is a type 3 CRISPR system. So there are other RNA CRISPR systems that have the problem that when they're activated, they also degrade other RNAs. But this particular one, this type 3 CRISPR RNA system, doesn't have this other nuclease that's going to just degrade RNA. And so you can do precise targeting of RNAs in theory with not a lot of off-target effects. Well, that remains to be seen. So that's what they're using in this paper.
Is it fair to say that all of these CRISPR systems are basically a protein RNA complex where the RNA guides the protein to a complementary sequence and there's cleavage?
That's right.
Yeah.
And beyond that, there's huge variation on that theme. Much greater variation than I was aware of before I read this. I said,
yeah, there's a lot. And there, because you look in various species of bacteria and you can get different enzymes and that have different properties. And furthermore, you can modify the enzymes to restrict and expand their properties as well. So it's a huge enterprise because people want to be able to do gene editing, right? That's what they want to do. So this type 3 CRISPR system in this paper, very interesting. It's made up of 5 subunits, which assemble around a CRISPR RNA. And there's an endoribonuclease that will bind to the target RNA and cut it at 6 nucleotide intervals. A very nice picture here showing how that works. So there's a series of proteins, including the nuclease, that will bind to the RNA. And you can direct it to a specific place on the RNA by using a target RNA. So they first establish that this works. They take the nucleocapsid gene of SARS-CoV-2 and they make the proteins. So they have to produce the proteins in cells and purify them and add the RNA that will target it to the RNA. And they show that it will cleave it with very high efficiency, and they make the fragments that are of the expected size. Actually, there's a very nice diagram in this supplementary figures where they show the RNA that they've made to target this protein complex to the target RNA and how the proteins line up one after another and cut that target RNA at every 6 bases or so. So it works. Now, here's the cool part that brings me back to the old days. When this enzyme, it's purified from Streptococcus thermophilus. Okay, so this particular enzyme, the type 3A CRISPR, is purified from there. When it cuts the RNA, it makes 2' 3' cyclic phosphates and a 5' hydroxyl at each cut site. And if you remember anything about DNA, that can't be ligated. DNA ligases need 3' hydroxyls and 5' phosphates. And in fact, when we used to ligate in the old days, phage T4 ligase, right? We used to buy it and use that to ligate, but you needed to have a 5' phosphate. And if you had DNA without it, you had to add it with polynucleotide kinase. All right. However,
good times.
There's a ligase from E. coli that can join the RNAs that are made by this CRISPR cleavage, the 2', 3' cyclic and the 5' hydroxyl. All right. So they try both of those. And to enhance the reaction, they make DNA. They call them DNA splints. The DNAs will hybridize to both ends of the RNA that have been cut and kind of hold them together while the ligases ligate the ends.
I thought this was very clever.
So they
Yeah.
basically came up with RNA repair.
Yeah.
They had to, right, because it's not going to be
Yeah,
repaired by the
there
cell.
isn't a good RNA repair mechanism. Let's build one.
Right. And so they found that, the both of these ligations worked, but the T4 was more efficient. So they decided to follow that. It's 25-fold more efficient, probably the smart thing to do.
And there's the paper's open access, and if you look at Figure 1, there's actually a really good, almost graphical abstract of what they're doing in the beginning of that figure.
So the basic way these experiments go, they will edit their RNA, and then they convert the products to DNA and sequence them, do deep sequencing on them, so they can see all the products that have been made. And they say we can make DNA splints that will capture different kinds of editing outcomes. So you can say if we want only 12 bases deleted, you could design your DNA splint to select for that, or 6 bases or whatever. So you could pick it. All right. So how do they actually show this is working? They pick Synbis virus, which is a plus-stranded RNA virus, 12.5 kb single-strand RNA genome, and it's got a green fluorescent protein gene inserted near the 3' end. So if infected cells are going to be green. So they designed a guide RNA. Remember, they're going to make an RNA which will hybridize to a part of the GFP RNA, and it's going to encode a protein, an amino acid that will disrupt the fluorescence. So they're
going to wreck the light bulb.
they're going to wreck the light bulb. So they're going to change one amino acid right, and it's no longer going to be green. It's going to be dark. All right. So they show that this guide RNA works on GFP RNA by itself, so they can get 6 or 12 bases excised, and then they take infectious viral RNA that they've extracted from cells. They incubate it with their nuclease, their CRISPR proteins and the RNA, treat it with T4 polynucleotide kinase to put phosphate at the 5' end, and then they put their DNA splint in and put in T4 RNA ligase. So they're going to cut it specifically and ligate it together. And then they transfect this bulk edited RNA into cells, and it's viral RNA now. So it's going to infect the cells and it's going to produce GFP, and they see a reduction in GFP when they've done the editing, consistent with, breaking the light bulb, as Alan said.
Yeah.
Everybody got that? So basically, They can edit the RNA and change an amino acid by this approach, which is pretty cool, which you couldn't do last week.
Yes.
Well, actually, they published this. Well, no, I guess that was still this week. So you're right. You couldn't do it
I'm
last
sure
week.
they could do this for the past, I don't know how long they've been working on it, right? But it's pretty cool. Okay, so then they say, well, what about deletions? Can we make deletions? Because here they've changed the single amino acid. And again, they've done it by putting an RNA in there with the DNA splint so that it gets ligated in. So now they do this. They again take viral genomes and do their CRISPR and do the deletions, and they then extract RNA from infected cells and they sequence them. And they have a DNA splint designed to make a 12 nucleotide deletion, and they do see that. 34% of the RNA genomes have that deletion, but they also see 18 base deletions, 24, 30, and 6, interestingly. So it's not perfect. These DNA splints are allowing a little bit of flexibility. Then they pick plaques, dear to our heart.
Yep.
And they get RNA from 11 wells that had CPE, that virus is reproducing, but they're negative for GFP, so they have been edited. 4 of the genomes have a 12 nucleotide deletion, which is what they selected for with their DNA splint, and the rest have a mixture of 12, 18, and 24 base deletions. So you have to search for the virus that you want. If you want a 12 base deletion you can't assume that they all have 12 base deletions by this technique. And maybe it'll get better. I don't know, but it's got a little bit of flexibility.
Yeah, I think it's interesting that most of the numbers they're using here are divisible by 3, so they aren't giving the frame shifts.
Yeah.
And I wonder what would happen if they use something that was 5, would they get a frame shift basically every time, whether it was 5 or 10, and that might be a little more complete.
You mean if they designed the DNA splint to do that?
Yeah,
right? Yeah.
to give them a 5 or a 10 deletion, even if you got half, it would still be of 10, you get 5, and you're still making a frame shift that way.
Right.
Okay, so the last experiment they do, can we insert a sequence into the genome? So far we've changed a single amino acid, right? But that's not the only thing we'd like to be able to do. We'd like to delete, which you can do with this. So you could take poliovirus and delete an entire gene. Of course, it wouldn't be infectious, but you could do that in theory. But what if you wanted to put a different gene in? We do that a lot with infectious DNAs, right? So they take a Synbis virus with blue fluorescent protein in it, and you can make it into a green fluorescent protein by changing one amino acid. And so they design an RNA, a piece of RNA, to make a single amino acid change, and they do their CRISPR cleavage of the RNA using a guide RNA, and then they have a synthetic DNA insert that has the single amino acid change that they put in there while they're using the DNA splint to do the ligation. And that they do this in bulk on viral RNA, and then they transfect this edited RNA into cells, and they find cells that are either blue or green, so it worked in a fraction of the cells, and they say about 5% of the bulk virus is now green. So they've edited about 5% of the virus with this technique. So I guess that you'd want to improve that, right? That's one of the things you'd want to improve, make it more than 5%.
Ideally, yeah.
And because I can imagine an experiment where you do this in an infected cell, right? You have an infected cell, you introduce this CRISPR nuclease and the targeting RNA and so forth, and you do it in an infected cell, and then you plaque out what comes out and look for the virus that you want.
Right, you do like a purification.
Yeah.
Hmm.
So you can alter RNA. This is pretty cool because I remember the motivation for us doing it in 1979 was because you couldn't modify RNA. So now you can, and I presume it's going to get better, right? Now that the basic technology is there, people will improve it, right?
Next week, you'll be able to order a kit.
Yeah, well, so what they had to do here was to produce the proteins and purify them to do these experiments. And they say, not everybody is going to be able to purify recombinant proteins or want to.
I smell a business proposition.
So yes, you could have companies selling them to you. Yeah. So I think it's cool.
Yeah.
And I think it's going to really contribute a lot to RNA virology.
Very cool. 2 days after the paper is published, we can see if those enzymes are already, for sale somewhere.
Yeah, that's right.
Says here,
But be careful buying them on eBay. You may not get quite what you expected.
we anticipate that in vivo RNA editing, so in a cell that relies on the cut and repair reaction, similar to those routinely used for DNA engineering, will emerge over time. So you could imagine, you want to change a specific, it doesn't have to be virus. It could be an mRNA in a cell, right? You want to change it and see what happens. So cool. I think this is very exciting. So good job, folks.
You'd have to use it as a tool in cells. There's a lot that would have to change. Like you'd have to jack up the efficiency considerably to see. Because if you do away with 5% or change 5% of the mRNA in a cell, I don't think you're going to see much happen.
You know what would be really cool there also is that it would be something you could do for like a short period of time.
Yes.
Because eventually that edited mRNA is going to get replaced with newly transcribed mRNA.
Yeah.
So you'd make kind of short-term edits or be able to really temporally control things.
Yeah, you could inhibit mRNA synthesis for a while to see that. Yeah, there are drugs that do that.
And you might be able to dial it down in increments. So actually, now that I think of it, maybe 5% reduction would be useful if you could also do 10, 15, 20, 90.
Yeah.
So while you guys have been yakking, I've been lost in the weeds here, and I want to relate some of that to you. I'm impressed with it makes me drool, the expression and purification of this enzyme.
Yeah.
Because as I understand it from looking at the methods, they create 4 different plasmids that go into a single bacterium, single bug, okay, that together express all 5 subunits of this thing, plus the guide RNA, which then assembles in this bug, and it's his tagged. Okay, so they do an affinity purification using a his tag, followed by a Cepheidex column or a superose column, so size exclusion chromatography, and come out with something that is pretty squeaky clean. All right, which just I can picture myself doing this and being really excited. But they make the comment, because this is one of the reasons I wanted to look into this. They make the comment at the end that with the current protocol, it requires expression of a new STHCSM cRNA ribonuclear protein complex for each new target. So as I understand it, they actually have to express the guide RNA along with the subunits and have them assembled in the bacterium and purify the whole thing for each target.
Yep.
But then they go on to point out, go on to point out that there are other CRISPRs out there where you can assemble an enzyme with a guide RNA in a test tube, and that greatly simplifies the process. But they aren't there yet with engineering those guys to do this. This is sort of proof of principle, and they're pointing out that there are probably down the road refinements that are going to make it better.
Yeah, because you'd like to sell the protein and then add your RNA.
Exactly.
Exactly. Yeah, that's going to happen, I'm sure. Where there's a will, there's a way. But yeah, this is good biochemistry here, Rich, and
I love it.
A lot of phage proteins involved, right?
Yeah, if I had it to do over again, there would be more biochemistry in my life. The problem is that biochemistry actually requires imagination, and I was lacking in that department. I let the virus do the thinking for me.
So I just for the latest Immune, which we recorded last week at Cornell, our guest was Ellen Rothenberg, who now does immunology, but she was a postdoc with David, and what she did was to take Maloney murine leukemia virus and have it make DNA in vitro, and she showed that the DNA was infectious. Okay, so she cracked open the virus particles and had them reverse transcribe the RNA genome. And I said to her, "Why did you do that?" And she said, "Well, nobody had done it, and we wanted to prove that, we knew that these viruses went through a DNA phase. Nobody had done it." And she said, "Nobody bothered to do the biochemistry to optimize the reaction." And she said, "That's all it took." And I said, "Well, David was a biochemist." She said, "Absolutely. And that's why it was the perfect lab to do it. He was a biochemist, and he said, optimize this reaction," and she got it to work. So biochemistry is great.
Yeah.
All right, let's do some email. I think, Rich, you should take this first one.
Okay. Ken writes, "Hi, Vincent and crew. Thanks for the shout out in TWiV 1039 and love to hear about bacteriophages on TWiV. In parentheses, I finally met Jolene in person at the Viruses of Microbes meeting in, what's that? Tbilisi, Georgia in July. The meeting was co-hosted by the Eliava Institute who have been researching and performing phage therapy for over 100 years. Nina Chancivelli would be great to have on TWiV, and parentheses. The reasoning for using archaeal viruses instead of phage or archaeophage is to emphasize the differences between bacteria and archaea. As far as I know, the person most responsible for this was the late Wolfram Zillig, who was a pioneer in the isolation and characterization of both archaea and archaeal viruses. Wolfram was an early and vocal supporter of Carl Woese and archaea being different organisms than bacteria. Carl Woese, the way that reads to me is Carl Woese is a different organism than bacteria. Yeah.
Yes.
Yeah.
Yes, that's fair.
Full disclosure, I was a postdoctoral fellow with Wolfram at the end of the last millennium. Cheers, Ken. And this is, we can shout out Ken. Here's Ken Stedman from Portland State University, where he studies extreme viruses.
So I this is interesting because remember we had a discussion with Jolene whether to call them archaeal viruses or archaeophage, and I thought viruses would be good. She said, so do I, but you and I, were raised working on eukaryotic viruses, and so we're biased. But this is a good reason to do it as well, right? To emphasize those differences, which are big. Yeah. And I thought you should read it, Rich, because you're a phage guy, basically.
Yeah, that's it's interesting. I get it now. I'm a little slow here, but you call them phage or archaeophage, it implies that the archaea are bacteria-like, and that's not true.
Yeah,
right.
Yeah. Angela, take the next one.
So, Salvatore writes, "Dear Dr. Angela Mingarelli, don't do freediving. Not even for fun. The documentary itself specified that the lungs are squeezed to the size of a human fist from the multiple atmospheres at depth. This is unnatural. The body was not made for this. And did you notice the thin body types? That is also likely a part of the repeated pressure at depth. The documentary scared me, and I was not even in the water, but I knew what they were talking about, particularly when, as you said, the diver reaches about 40 to 60 feet and you begin to sink unless you adjust your BC, body composition, I'm guessing. Yeah.
Buoyancy compensator.
Buoyancy compensator.
Oh, buoyancy. Okay, sorry.
Flotation vest.
Which is also, well, you don't wear. Okay, I'll finish.
Yeah, that's a scuba. It's a scuba thing.
Yeah, you're floating in space, but you must keep your eye on the depth meter. And thank you for all your input on the show. When is the best time to visit Azores? Been listening and learning since March 2020. The show was a light in the storm of misinformation. Also, thank you for occasionally explaining to us laymen when the show gets into the weeds of science. Sincerely, Salvatore, Philadelphia trial lawyer for over 50 years.
Cool.
So thank you for your email, Salvatore.
So Angela, what's the documentary he's talking about? Did you mention?
So this is the what is it? The last breath? No, the deepest breath.
Okay.
Sorry, the deepest breath. I'm sorry. It's the deepest breath on Netflix that we spoke about on 2 TWiVs ago when I was on.
Right.
So I think that when he says that it's unnatural for humans to dive, technically we have adapted physiological mechanisms that are meant, like the humans have, that other mammals have for diving. It's called the mammalian dive reflex. It's part of the vagal nerve, in your face. If you put your face in cold water, it automatically causes bradycardia and your blood starts shunting to your vital organs. So
wow.
I do very recreational free diving. He doesn't have to worry. That movie is very hardcore professionals. These people only free dive. They're going down 100 meters, so over 300 feet. I don't do any of that. My maximum free diving depth is 28 meters. And that's what is that? Almost 90 feet, I guess.
90 feet, yeah.
Which is very recreational. It still takes some skill, obviously, but I'm thank you for your concerns, Salvatore. But also in freediving, you don't wear any sort of buoyancy jacket. That's in scuba diving. In freediving, you just wear either a wetsuit or nothing at all, and weights. So there's no I thought he was saying body composition, because it is true that if you have more fat, you float more, and if you have more muscle, you sink more, just density wise. Yeah, visiting the Azores. I didn't live in the Azores. Those are another archipelago north of the Canary Islands, part of Portugal. But the temperature is probably similar. I would say the summer is nice. It's very green. It's kind of like the Canary Islands, but greener. Um, and yeah, the thin body types due to pressure also not physiologically kind of that it's mostly just because if you're thinner and you have less body fat, then you'll just sink faster. You won't see people like fairly overweight or that have like larger compositions normally free diving, but it doesn't have to do with the pressure. It just has to do with like being more effective in the sport. But yeah.
So
I would just add one more thing to this. There are groups of people around the world that have been freediving for hundreds, perhaps thousands of years. There's an ethnic group in Japan, I think, that's been doing this for centuries, diving for food and for pearls, and I think in parts of Southeast Asia as well. And they have traditions built around like how long you stay down and, safety protocols essentially. And these people have been doing it for generations, and yeah, it can be done safely.
Yeah, and some people actually have they've adapted like physiological mechanisms like with their eyes where they can actually see better underwater, certain populations. I believe they're the Japanese that you're talking about. Or there's also a Mexican tribe that also did this where they can actually see better underwater because they've been diving for thousands of years. And I'm not sure if it was like their pupil or like their lens, like it's in a different shape that they can actually, like most of us, when you open your eyes underwater, it's blurry, but they have like a more acute vision underwater.
And it may be either inherited or environmental. Like if you start diving when you're 5 years old and you do this your whole life, maybe your eye just grows differently.
Yeah. So definitely you have to be careful free diving, which I wanted to give emphasis in our last episode. If I didn't, then I will emphasize again that you should have training to do this. It can be dangerous, but that humans do have, like Alan's saying, physiological mechanisms that allow us to dive that literally slow your heart rate down so you consume less oxygen. Like the more you dive, other mammals, marine mammals have high myoglobin levels. Like mammals are very adapted to diving more than we think. So, yeah.
All right. I thought it was interesting how he was concerned for you, Angela.
Yeah.
Yes.
Good.
I appreciate his concern.
Don't do free diving. Okay. Take it back.
Brienne, can you take the next one?
Sure. Larry writes, RE free diving with nanobodies and interferon. And this is a not biotrained retiree listening since Ebola. I was worried about the source of nanobodies from camelids. The sacrifice of the monkeys makes me cringe, although I am grateful for them. I asked Google's Bard, so I am not sure how correct this is, but it sounded like no sacrifices required. Maybe not correct? And Larry is from the Midwest. Yes, Larry. So typically we are using genes that came from camelids that encode antibodies, but we have them in cells, and we are doing this entirely synthetically and with recombinant processes.
Well,
and all you need to get the genes from the camelids is a little bit of blood.
Right.
Yeah.
But you do have to immunize them if you want specific nanobodies, right?
I guess you would at this point for a camelid.
Yeah.
For others,
take serum,
for other,
right? Yeah, you just take.
Yeah. For other organisms, we've got it to the point where we have cell lines, and you don't even have to use the animal at all. But I don't think we have those camelid cell lines.
Yeah, and just economically and technically, that is something where you would definitely just take blood samples periodically from the same animal and purify serum.
And they keep the camelids and use them for other antigens that may be different. That sack. It's not like a rabbit where they would sack it after one, because the camelids are very big, right? And you don't want to be right, Angela. You don't want to be
and very expensive.
Them.
No, we actually have 2 dromedary camels at the veterinary faculty where I was in Spain, and they were doing MERS at this point. They were doing MERS studies, and they would serially take serum from the camels. But you definitely would not want to go through camels as fast as we do some mice, because it wouldn't be feasible.
Well, I can imagine also the amount of blood you could get from a camel and not
significantly larger than what you can get from a mouse
is quite a bit more than you could get from a mouse.
More than 100 microliters for sure.
I think.
Yeah, I saw somewhere they were doing MERS experiments, and camels, and they had a young camel, and they had a mask on its face. I felt very bad for it, And eventually they had to sack it, because they wanted to take lungs out. So, MERS
When we
is
say sack, we mean sacrifice. For those that don't know what that means, it's kind of euthanize, exactly.
Yeah.
But sack is short for sacrifice, which when I first learned that, I thought it was like a little aggressive, but we become so used to saying it, like sack the mice.
I think they don't prefer. We prefer.
That is no longer the preferred term.
Yeah,
we say sack though. Unfortunately, we do, even though like we try
Yeah.
to use the word youth, but we end up saying sack or take
Yeah.
down. Some people use the word take down as well, but
yeah, I feel badly for the camels though. But
yeah,
most of the time.
But for the nanobodies,
need
that
to,
is
yeah.
not necessary.
That's right.
You only have
Camels
to
are
do
okay.
that if
The
you
camels are
if you
okay.
need like an organ to analyze that.
Yeah.
What do you feed a camel?
they eat mostly grains, I believe. Like whether ruminants, well, they're like a weird type of ruminant. They're an adapted ruminant. Like they have like 3 stomachs, I think, from what I can remember. But mostly like other ruminant species, grains, etc. Hay.
Hey,
Yeah.
Cool.
Do you ever get to see the camels, Angela?
Yeah, actually, when I went to Spain this summer, I went to the campus to show my partner because he hadn't been there, and I was like, "Oh, we have camels!" So I went to take him to show him the camels, and they were great. They're awesome. We have 3 of them now. Actually, before we had 2, there was a third one. They're a little bit like, I want to use the word sketch, but like you can't really get too close to them. They can get a little bit uneasy around people that they don't know, so you just keep your distance because camels do spit. That's a real thing.
Oh, that's what you're worried about, them spitting at you? Yeah.
Yeah, and like the fence that we have, it's kind of like just to like chest height, but it's not really a fence. It was just like a bar. So camels have very long necks. They could like reach and
bite you
or anything like that. I don't want to be bit by a camel.
I once had an interaction with a very aggressive llama that is really impressed me with. Boy, I don't want to do that again.
Yeah, llamas and camels are not as friendly as they seem. Having worked with them,
well, Rich, it's tough out there in the nature. They have to defend themselves.
Yeah. I mean, I don't blame her. I was in her house. She said, "Get away from me." She let me know.
Alan, would you be interested in taking this next long one?
Sure, I will take John's letter. Hey, TWiV podcast. I'm John, a new listener from Mississippi. My pediatrician mentioned your podcast, and I decided to check you out, and I've been pleasantly surprised with how interesting, informative, and accessible your podcast is to a layperson with a cursory science background. Bachelor of Science in Physics, computer programmer. Wife has a Bachelor of Science in Biology, quality control and pharmaceuticals, CSI, seafood import. So much cooler than me. I wanted to comment on the loss of confidence in the institution of medicine, because I suspect you would attribute that largely to vaccine information and the RFK types you so handily debunk. I just want to give voice to the less talked about and more powerful explanation for this distrust. Because despite having nothing but respect and admiration for scientists and doctors, I have a lot less faith in our healthcare system as a whole these days, and that is because our healthcare system sucks. The root cause is quite simply capitalism. I haven't heard enough of the podcast to know how often you get political, but I'll quickly lay out my case. Okay, so just a quick one, John. If you go back a little bit, you will find that you are talking to a receptive audience on this topic. And this is our discussions of loss of confidence in the institution of medicine have actually touched on a number of aspects of it for different groups of people, and also not just from the vaccine side, but the vaccine issue is kind of that's our turf. So that's what we talk about most. And then goes on to explain we have worse health comes in the U.S. than Cuba. Just stop and think about that for a second. That is an absolute embarrassment. The richest nation, which spends 4 times more on healthcare than its European counterparts, has worse health outcomes than a communist country we have demonized and cripplingly embargoed for decades. We have a decreasing life expectancy due to deaths of despair, and that comes before COVID hit. Now, my personal frustration. I just wish I heard more doctors comment on this. Okay, you've never been to my house, John. I hear one particular doctor comment on this on almost a daily basis. In fact, I was chatting with her on the phone on our drive to work this morning about things related to this exact issue. So John continues, every time I convey this frustration to a doctor here in Mississippi, hoping to elicit a response of solidarity, because after all, we are both stuck with this this bad system, I get some recycled talking points about how actually universal healthcare isn't as good as our system with anecdotal evidence in support. If our healthcare system is so much better, we wouldn't have worse health outcomes. If it's better for the people who have access to it, but when including those who don't have access, the aggregate is pulled below countries with universal health care, then that really ought to give us pause. Do we want to endorse such a caste system where your health and value as a human is entirely dependent on how much money you have? At this point, even conservative think tanks like the Rand Corporation are publishing studies that say universal health care would save us trillions of dollars. It seems like doctors' ability to think systematically evaporates when considering economy or politics. And then discussion of systemic thinkers and how they would approach this differently. So here's my call to action. Doctors have to lead the way on this issue. Doctors know better than anyone how stratified health outcomes are if they merely pay attention. Doctors have to sound the alarm. It makes your pushback on people like RFK more credible when you can acknowledge the healthcare system has become so bloated with costs and exploitative it is reasonable to feel disenfranchised. People are disenfranchised. We need Medicare for all. Maybe you could loudly support physicians for a national health care plan every episode until we get there. pnhp.org. Okay. Rant over. I hope I'm talking to a sympathetic audience. John, you absolutely are, at least from my own perspective and from what I know from what other hosts have commented on in the past. As I say, this is something that comes up frequently in my household, but in addition, I think your interactions with physicians may have been skewed by the state that you're in. The physicians that I have talked to about this subject, and that's been quite a few over the years, have pretty much condemned the U.S. system as unsustainable and exploitative and inhumane, and all sorts of other things that I can't use the terms for on TWiV. And yes, this is a huge problem. And there was tremendous resistance to even making the change that we initially needed, which was Obamacare. And I don't see a whole lot of hope for this getting fixed in the near future, but hopefully in the long term we can eventually get there.
I mean, I hear horror stories about people in Canada who have to wait and they don't meet criteria for procedures.
There was this one guy who had to wait for, okay, well, how about the millions of people in the U.S. who just don't even get in line for that operation because they can't afford to see a doctor?
It's true.
Well,
that's the problem with the anecdotal evidence is it's very powerful because it happened to somebody in particular, but it is very weak statistically, and when you look at the overall picture, you see that pretty much all of the other systems in the developed world are far better than ours.
Well, I know a lot of Brits who complain about their health care as well.
Yeah, but they haven't lived here, have they?
I'm just saying what I hear, that's all. I mean,
yeah.
If you're going to make it, you got to make it work. I mean,
of course.
Obamacare has helped a lot of people.
Millions,
and,
yes.
Apparently Medicare is very good also.
Yes.
Hey, Angela, do you have any impressions of the healthcare in Canada?
I mean, I think that we're, compared to the United States, definitely privileged if we compare ourselves to the US, but I can go at any time to the emergency room and maybe I'd have to wait. It depends on the severity of what's wrong with you, right? But if there's something severe, like if I were to have some sort of like hemorrhage or like some sort of bleeding that needed to be controlled, I would be taken almost immediately. But if it's something like a few months ago, I split open my chin and I was thinking about getting stitches, but it would have been a 12-hour wait because it wasn't something that serious. So in the end, my cousin is a nurse and I called her and we just triaged and I got a butterfly like Band-Aid and I did it myself. But it's like Alan was saying, it's 12 hours as opposed to just not getting attended at all. And that's for something that's not very serious. So I think that we're definitely better off than the United States. That the waiting times are longer than they used to be a decade ago, yes. But that we still have access to very good healthcare.
And I will
And free.
point out that
Well,
there's
free.
Yeah.
there's a non-zero number of emergency rooms in the U.S. where you could wait over 12 hours for a more serious issue than a little
Yeah.
cut on your chin.
So,
Yeah.
Alan, what,
and pay a lot more than free.
And pay a lot more than free. Yes.
Alan, what is the chance that we will ever have that in the U.S.?
I'm not going to give odds on politics, but I think in the current environment, just nothing is getting done.
Yeah.
The system's paralyzed with this stupid nonsense that's being promoted by one side to drum up discord.
All right.
John's got a PS here.
Oh yeah, John says, I wonder if y'all have read Michael Lewis's The Premonition on the U.S. response to the COVID pandemic. It wonderfully illustrates these problems with a more intimate view of the more systemic problems. It paints an obvious picture of how 50 disparate and unique healthcare systems leave us woefully underprepared for the cooperation and resource and information sharing required for a pandemic. Yes, I think that was somebody's pick a little while
I
ago.
definitely have read it.
Yeah.
Michael Lewis is the Moneyball guy, right?
He is.
Did you like it, Brianne? Was it good?
I did. I mean, I read it right when it came out, so it's been a while, but I did
right.
like it.
So thank you, John. This is an important topic. And
Yes.
So that's good.
Definitely.
All right. John writes, Doctors TWiV, I thought you might like this. If the image doesn't transmit, let me know. I'll copy paste. Rob Rogers, as you may know, was the, what's that, Pittsburgh
Pittsburgh.
Post-Gazette's outstanding cartoonist until he was sacked by the uber right-wing Block family owners of that paper that Pittsburgh could once be proud of. Because of that, I refuse to subscribe to the PG anymore. Meanwhile, Rob is now freelancing and his patrons get his cartoons. How right-wing is the Block family? Have a look, and he gives a link. I guess there was supposed to be an image, and it didn't transmit, and I'm sorry I forgot to ask you by the time I put this here. Meanwhile, here in Greater Braddock, the heat wave broke yesterday. I think that event is just reaching you now. So the next two emails are both about the USAID terminating its wildlife virus hunting program, which everyone's certainly heard about, right?
Mm-hmm.
Yep.
Unfortunately.
Yeah.
Deep Vision, it's a $125 million project for discovering and cataloging pathogens from around the world, was ended, a virus hunting endeavor, because the officials said it raised the risk of an accidental outbreak. I'd like to see the data for that.
The first sentence in the first letter from Alan is just kind of stunning. If scientists don't go and look for a virus in the wild, the public cannot be harmed by it. Do the viruses know that?
Tell them to stay in their lane.
Yeah.
U.S. Army Major Walter Reed.
If you hadn't turned on the headlights, we wouldn't have hit that light pole. You wouldn't have seen it at all. Yeah.
Walter Reed and William Gorgas must be turning over in their graves at this foolishness. Yeah, I mean, this is a political decision, right? To appease certain people who say we're doing dangerous things.
Yeah.
And it's really unfortunate. We need to see what's out there. So we had a letter from Alan and a letter from David, who both, well, so David wants to know our thoughts on this. And I think it's absolutely absurd to cancel that program. I think we need to know what's out there. And there is zero evidence that sampling wildlife is dangerous. What do you think, Angela? Is there any evidence that we've started a pandemic by sampling?
Well, definitely not. I think that the only way people could see that it could be dangerous is that scientists that are unprepared, which isn't the case, because those that go sampling are very well prepared, use all of the PPE, personal protective equipment necessary, have all of the samples with all of the permits necessary, import permits, export permits, wherever they're going. It's very controlled. I think that some of the people that have these opinions just think that we're just running out into a cave and grabbing a bat and like killing the bat and then taking it to the lab. Like that would never happen. These are all very controlled environments and they're extremely regulated, like all of these sampling efforts. We've been trying in my lab to sample bats for a while, and it's very difficult because the Canadian Food Inspection Agency, like the CFIA, to import those samples, it's almost impossible. Even if we inactivate them, we show them inactivation, like it's very difficult. So I think that that just those that are sampling are very well qualified. The scientists that are doing so, we just have to remember that, and that they know what they're doing, and they're very like thought through protocols that maintain ourselves safe. Because obviously, let's say if I'm in a cave sampling bats, I don't want to get any sort of, I don't know, Nipah virus or something that could be in that bat.
Or rabies.
Or rabies,
Yeah.
exactly. Well, I would have to get prophylaxis
You'd
to
be
rabies.
vaccinated for that, but yeah.
But yeah, other things.
Yeah, it's not as though every interaction with animals has all of that PPE. This is in fact a rather safe interaction.
It's very safe.
Yeah,
exactly.
And remember that this is wildlife. This is out there around people who are not wearing any PPE, who happen to go into that cave because it looked cool to explore, and they're wearing, whatever they were wearing, or who happen to be, working on their job at the farm, and the bats are roosting in the attic above the barn where the pigs are, to cite a Nipah virus example. That's how these things happen, and that is going on every day, that people are having those unprotected interactions with wildlife. So somehow the notion that scientists who know what they're getting into and are going in like gowned and masked are somehow a greater risk, that's just absurd.
It's ridiculous. But hopefully other countries continue with their efforts, and that's all we can do.
Well, one of the papers I was thinking about for this week is a big bat sampling study out of China, right? Completely China-driven bat sampling, different species.
Good.
They're doing it, which is great.
That sounds awesome.
Yeah.
Escape the silly politics, as Alan said, of this country. Everything is paralyzed. It's ridiculous.
Yeah.
I think it's because a lot of people are still so convinced, even some politicians, that the lab leak theory is very valid. So they would say, why would we sample animals anyway if it was from a lab anyway? It was fabricated in a lab. Right. Which is ridiculous in itself. But I mean, I think that is a large part of it as well, that they justify the not sampling animals with that. But it's unfortunate.
Someone tweeted when this was announced, oh, great. Now this will reduce chances of future pandemics coming from laboratories, as if we had one already.
Right.
It's very difficult to listen to all of this personally. I just get very frustrated. I'm sure many people do. All right.
Yeah.
Thank you, folks, for your emails. Send them in. TWiV@microbe.tv. Let's do some picks of the week. Angela, what do you have for us?
This week I chose, first it started off as a Guardian article from The Guardian, but then I actually went to the publication itself in Current Biology. The publication, the paper is called How Many Metazoan Species Live in the World's Largest Mineral Exploration Region. So basically there's this region called the Clarion-Clipperton Zone between Mexico and Hawaii. It's about 6 million kilometers squared. But it's very rich in minerals like cobalt, nickel, copper, etc. So China and the United...
On the seafloor.
Exactly, on the seafloor, sorry. And there are certain countries that are very interested in mining this area. But this group specifically, the group of... So Muriel Rabone is the first author. And they found over 6,000 new species in the CCZ area. This they call it the CCZ, the Clarion-Clipperton Zone. So they found over 6,000 new species, and they're trying to, I think, now there's a little bit of like, we're in a limbo where if these are endangered species or not, if they're going to be able to mine or not. And I thought it was just amazing, like apart from like the politics in this, that they found 6,000 species in this area. This is like obviously it's a huge part of the ocean, but relatively small-ish. Like if you look at the whole Pacific and Atlantic, it's fairly small. And that they found 6,000 new species. I think that's amazing. Like there's so much on our planet we still don't even know about in our oceans.
How did they, is this just by looking? They didn't do metagenomics, or it was all metagenomics?
Let me see. I didn't actually...
They got pictures of these creatures.
Yeah, they do, which means that they must have actually, let's see.
Must have pulled them up.
I don't see any sort of like metagenome in their figures. They just have they don't have any metagenomic analysis. It's more so sampling effort.
Look at that. That's impressive. Wow.
I don't actually know how they sampled. I didn't get that far into it.
You
Yeah.
got some cool looking critters here.
Mm-hmm. Yeah.
They just say sampling.
I think this may be a data mining paper.
Yeah.
Yeah.
Because they
Which
show
is
the
fine.
specific regions, like geolocated
Yeah.
published records of metazoan literature and databases. Ah, true. You're
Yeah.
right, Alan. You're true.
Yeah. So they're searching. There have been lots and lots of expeditions. I mean, the reason we know there's cobalt and all this other stuff
Mhm.
there is because of expeditions that have gone to these places and sampled a bunch of things. And I think in a lot of these cases, they get so many samples and so much information that they just dump it in the database and say, yeah, we'll analyze that later. And so these folks are going through and building what they call a checklist of species in that zone. So here's what's been seen so far in just these sampling expeditions.
Yeah, cool.
Yeah. So I'm sure
right,
there's
ne-
a lot of metagenomics in there. This is just, as Alan said, like
Yeah.
a agglomeration of all of it.
I'd
But
like
it's
to-
really cool.
Yeah.
I'd like to
Yeah.
grind them up and sequence them and get some viral genomes out of them.
That's true.
But-
I'm sure there are many. How many millions of viruses or virions are there in 1 milliliter of seawater,
Go ahead, 10 million.
Vincent? 10 million?
10 million.
10 million, the population of New York
Crazy.
City.
That's crazy.
It's a lot. Yeah. Well, that's coastal seawater. It's less when you go deeper, but still, it's a lot. Yeah.
I mean, I've drank a lot of seawater accidentally while surfing or swimming or whatever. So I've got all those aquatic viruses.
Well, they pass through.
Of course.
Maybe you got some
Maybe of them
nutrition
cook.
from them. Yeah, you got some
Yeah.
nutrition from them. That's a lot of viruses. We did a paper where they showed, plankton can exist on viruses. Remember that, guys?
Yeah.
Yeah.
All right, Brian, what do you have for us?
So I have something that a physicist friend of mine posted somewhere, and I just found it really fascinating. So this is an article from the New York Times talking about some of the issues in cosmology right now, specifically thinking about some of the observations that have been made with the James Webb Telescope and how they challenge some of our basic assumptions and basic knowledge about physics and may require rewriting some laws of nature that we didn't know could be rewritten or think about. At least I was reading and I'm like, wait, what? So they talk about how
We thought there were laws. They turn out to be guidelines.
Yeah,
Yeah.
I mean, one of the
Yes.
things that they mention is that perhaps the rules of physics are different in different parts of the universe.
Ooh.
And so perhaps the reason why some of the details of expansion don't match up to what we think is because we're looking in places where the laws are different. And they're sort of hypothesizing on how can they square some of the observations that they have that don't make any sense, and how they have to almost rethink assumptions and almost start to rethink about the philosophy behind some of these things of what assumptions are they really making and what are they philosophizing. And I just found it very interesting. It reminded me of some sci-fi I'd read recently that argues about some of those same things. And I was fascinated the whole time I read this. So
Very cool.
Cool.
Sounds like uncertainty principle.
Uncertainty principle, much, much more. Uncertainty principle on steroids.
Yeah, that's true. Rich, what do you have for us?
So, my wife and I are usually up drinking tea in the living room around sunrise time, which of course changes during the year. And we were making the observation recently that it seems to change faster this time of year than other times. And I've noticed this before. We've talked about this before, and I've never been clear as to whether it actually, the rate of change in the length of the day is different at different times of the year. I never really thought about it much, or whether I'm influenced by the fact that, at different times of the year, I'm up or not when this change is happening from night to dawn. And so maybe I'm more observant. So, of course, I Googled it and I found a little Scientific American snippet, basically, that says, yeah, the rate of change is different at different parts of the year. And it also depends on what latitude you're at. All makes sense if you stop and think about it. This has nice little charts that talk about this. And where we are at 30 degrees north this time of year is, approaching the equinox. is about the fastest rate of change. It's up to 2 minutes a day. So over a week, you can have 15 minutes difference, and that's enough. That's enough to be noticeable. Different times of the year, it's the rate of change is near zero. So there you go.
Cool.
Very cool.
I don't like. It's already dark now. I don't like this.
It's not dark yet, Vincent.
It's not dark.
Well, in my room it is.
Yeah, but you don't have windows in that room.
Okay.
That doesn't count.
Oh, yeah, that's different.
Well, 4:39. At some point in the future, it's going to be dark.
That's true.
Yes, that is fair
December?
to say.
I think it's like December, around
Yeah.
Christmas, right?
I
Is
don't like that because then I leave, it's dark,
Yeah.
and in the
Oh
morning. I
yeah,
leave, it's
you know,
dark.
when we used to live in Buffalo, you get up in the morning and go to work, and it was dark, and you'd come home and it was dark.
Mhm.
Yeah.
Oh well. Alan, what do you have for us?
I have something that also grew out of a conversation with my wife. So good transition, Rich. Thanks for setting that up. We were a few weeks ago, we were on vacation in a couple of different European cities, Madrid and Barcelona, and then we went to Mallorca. And by the way, highly recommend all of those places. Amazing trip.
I concur.
But yeah, we were doing a lot of walking around because, you know, European cities, great transit systems, and you walk to things. And at some point, Laura said, she was walking a little behind me. She said, Alan, when you walk, you swing your left arm further than your right. And I mean, one of the things I love about her is that she has a keen sense of observation, but sometimes it can be a little awkward because, of course, immediately I was almost paralyzed by this. You know, oh, God, okay, I'm
Stiff
gonna
arm
swing. I'm
walking.
just going to put him in my pocket. Yeah. So now I should have warned people that once I discuss this, what I'm going to describe, you will not be able to unsee.
Oh no. Unhear.
Yeah. So, after, you know, giving up on trying to control my arm swing, I was like, okay, fine. It's just an asymmetry that I have. And then my daughter, Sophie, said, oh, that guy does the same thing. And there was a guy walking down the street ahead of us who his left arm distinctly swung a little bit further than his right as he walked. And so we all started looking around at everybody walking on the street, and we noticed that almost all of them had a wider arc on their left arm swing. I mean, discounting people who had something in their hands or, you know, hands in the pockets or whatever. Everybody who was walking with their arms free, the left arm swung through a wider arc. There were a couple of people where it looked almost symmetrical, and I think there was one person where it might have been the right arm further. We did not run up to them and ask them if they're, you know, left or right-handed.
Yeah.
But partly because our Catalan isn't very good, but also it would have been a little awkward. So anyway, I, of course, went to PubMed and said, is this a thing? And yeah, it's a thing. And what I'm linking to here for my pick is a Scientific Reports article from 2018, which appears to be the most recent research on this. It's by a team of neurologists. And they're, of course, very interested in how people walk, because that's diagnostic for a lot of stuff. And they talk about the findings of directional and non-directional arm swing and asymmetry in arm swing while walking. And it's just a neat paper. It's open access. And it turns out the majority of people, they looked at like 300 people in this paper, and they had them walk down a hallway with wristbands with accelerometers, kind of like your Apple Watch if you have one of those. This is a very cheap way to assay arm swing, and they found most of the people had a distinct left arm preference, even the lefties. They only had a few lefties in the study, but they also swung their left arm further. And they talk about that. And they did a cool thing where they had them walk normally, just walk down the hall. And then they gave them a task to do. Okay, walk down the hall. It's a serial subtraction task. Like count backward by sevens from 397 or whatever, you know, some weird number. And then you've got to think about that while walking down the hall. And they found that reduced the asymmetry.
Interesting.
Now, what that means, it's unclear, but I thought this was just a really cool phenomenon.
I am now going to be very, very paranoid when I'm walking home and observing my arms.
Yes.
Me too. I'm
Everyone's-
left-handed. I wonder if it's
You
true.
can't unthink this once you know it. And every time I'm walking somewhere, I'm like, oh, yeah, left arm further. Oh, that one's left arm further too.
I wonder if the right arm is favored in the southern hemisphere.
Yeah. Yeah.
I mean, why? I mean, the
I don't-
left-handed thing is the most obvious, but that obviously doesn't do.
That doesn't seem to be the answer.
Sometimes I walk with my hands in my pocket so you don't swing it, you know.
Well, right, yes.
So, is there any correlation between handedness and left brain, right brain activities, or is left brain, right brain independent of all that?
I think that's been kind of debunked or
Not
partly
so.
debunked. There's not really a whole lot to the left brain, right brain
Oh,
thing.
so
I
I
mean,
have
they-
to forget
yeah.
all that?
No, I mean, they do. There is a distinction between the two hemispheres, but it's not nearly as, it doesn't affect nearly as much as
Okay.
was
I
once
being
promoted.
left-handed, I remember when I first got to the lab after vet school, which I didn't have a lot of lab experience. So I had to I had pipetted before a bit, but I was like, okay, what hand am I going to use? This is like a moment that I need to decide.
Yeah.
And then so now I started using my right hand to pipette, but now I can also use my left hand. And I always,
Ooh.
this whole hemisphere thing, I thought it was more true than it is, I guess, because I'm always forcing myself to use my left hand and my right hand because I'm always thinking I'm going to use my whole brain. And I'm always like left hand, right hand. I try to
Right.
open the tube with my right hand because I open my falcon tubes with my left hand. Sometimes I'll try with my right hand. That's more difficult, the dexterity for opening the falcon tube. But yeah.
Neat. Well, that's quite interesting. Now, I'm not going to want to swing my arms at all.
Yeah.
I'm not going anywhere.
Just
staying
sit
right
in
here.
place.
We're all
Well,
traumatized.
I haven't swung them for the last 2 hours, so that's cool. All right, my pick is They Farm at Javits Center. This is really going to be for Dixon, because I just learned about this. But here you go. Javits Center is a convention center here in Manhattan. It's not far from the incubator, and it turns out there's a rooftop farm, which was put up in 2021. And this is a website which describes it. An acre of green roof vegetable growing beds, 3,500 square foot greenhouse, and a 10,000 square foot rooftop orchard where they have
Cool.
38 apple and pear trees.
That's heavy for
Wow.
a
Yeah.
roof.
Yeah, that's a
38
lot.
trees.
Yeah.
Wow.
And then they have a mobile garden near it with 100 movable planters with perennials. So this is on top of the Javits Center.
Well, they're apple and pear trees. They're not like oaks.
But some apple trees are very large. At least I go apple picking.
Some
around
apple
here.
trees can get big, but the
Huge.
ones that are commercially cultivated tend to be
Yeah.
kept.
So this is done by
Sure.
an organization called Brooklyn Grange, which has farms in Brooklyn and Queens as well. And they were contracted to build this one on top of the Javits Center. And so you can go see it and visit it. But the website, they have a nice diagram of the circular systems, right, that recycle a lot of things. For example, in the greenhouse, all the food waste is composted and used,
cool.
and it's added to the soil and so forth. So,
I like that they can collect and recycle the water that's not taken out.
Yeah.
Mhm.
yeah.
This is super cool. I had no idea, and I'm
Yeah.
going to be telling my environmental science colleagues as soon as we're done here.
Yeah, you should go visit it.
They probably will.
It's not far. I should go sometime and check it out because it's not far from me at all. I was very surprised. So someone told me this the other day, and I didn't know this existed. There you go. So many things you don't know. By the way, Dixon wrote a new book. He's probably going to
really?
pick it at some point. It's called the New City, I think, and it's all about.
I pre-ordered it. I don't remember what it's called.
Yeah, it's the New City. I have the cover right here. How to Build Our Sustainable Urban Future. And I interviewed him last week for it, so get him some publicity. I think it's publishing in November. But it's got all
Cool.
different ways that he thinks cities need to be made sustainable. And, one way is to have every building recycle everything, make its own energy, make some food, and recycle all of its water. And there's a building in San Francisco that already does that. So all of the wastewater is recycled and people drink it.
I mean, if it's like purified and made potable and all of that, why not?
Just don't tell me
think about it.
Don't tell me
No,
about it.
that is like something that NASA does.
Yeah.
I think routinely, and it is also done in some places such as the Galapagos Islands.
Oh, cool.
If you go to the Galapagos, you don't drink the tap water, but you can wash in it because they purify it to a decent standard and recirculate it.
Anyway, I'm sure Dixon will talk about that once it is published. We have a listener pick from Asal. Hi, Vincent and TWiV co-hosts. I'm sharing this as a listener's pick. Pretty sure Dixon would like it the most. The Ig Nobel Prizes.
Yes.
Honor zombie spiders, rock-licking scientists, and a clever commode. So this commode is the winner of this year's Ig Nobel Public Health Prize. The inventor sits on top of his invention, the Stanford toilet.
It is a smart latrine that can monitor an individual's health by analyzing their urine and feces.
And it identifies the individual by a combination of a fingerprint scanner and an anus scan.
Oh, wonderful.
Ooh.
Wonderful.
It recognizes the shapes of different individuals. Yeah.
So this is joking stuff, right? This is a joke, correct?
I mean, no, it's a
No,
serious
it's a real thing.
invention.
It's real,
that's
no?
to be used for
Yeah,
medical-
the Ig Nobel Prizes, it's real published research.
Yeah.
Yeah,
Why- so,
they're
but-
basically
but-
the idea is the prize is the silliest thing.
Oh,
Yeah.
I
It's
see.
real.
Okay. It's just silly, right?
Yeah.
Yeah.
Oh my gosh. And
Imagine
then
giving your
there's-
fingerprint and they'll be like, and we need another.
Yes.
Area.
There's one other biometric test.
Well,
Harder to
the toilet has a sensor
Mhm.
that takes a picture and scans, so you don't have to do anything for that, right?
No.
Yeah.
Interesting.
But even knowing that it's getting taken is a little
Just
like,
knowing that's
It's
yeah.
they have a name for it. It's an anal print.
anal
Yes.
print.
Then there's the Literature Prize for researchers who studied a phenomenon known as jamais vu, which is the opposite of déjà vu, when an individual perceives something as being unfamiliar.
But isn't that like everything that you haven't
No,
experienced before?
it's specifically they give an example in this write-up in Science that you can create this sensation in a lab by having somebody repeat a single word many times. Now all of a
Okay.
sudden the word
yeah.
many
Yeah.
sounds alien to you.
Yeah.
Sounds
Yeah.
weird.
Oh,
Yeah,
that's true.
I was originally
actually,
thinking it was the time when I tell my students, yeah, we learned about this before. And they're like, what? We never learned about this.
Yeah, that's right. You never told us this. Yeah, you give them a quiz on a test, and they say, you never taught us this. I get that a lot.
Now that
Mhm.
lectures are recorded, you can say, go check the lecture.
Yeah.
Before you could be like, did I say that?
So there's a lot of things that although the Ig Nobel Prizes make fun of some of these things, they know at the same time that there's legitimacy.
Yeah.
There's
to a
serious
lot of these
science
projects.
going on
I
here,
think,
yes.
in fact, if I'm not mistaken, the psychologist Dunning and Kruger, who characterized the Dunning and Kruger effect, got an Ig Nobel Prize,
I
and
think
that's
that's correct.
that's an important observation that's
Yeah.
relevant, highly relevant,
all the time.
all the time.
All right. All right, that'll do it for TWiV 1045. You can find the show notes at microbe.tv/TWiV. Send us your questions and comments. TWiV at microbe.tv, picks of the week. We like to get yours too. And as I said at the beginning, please give us your financial support. Go to microbe.tv/contribute. Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time. And for you Austin TWiV people out there, on November 11th, Veterans Day, Acapella, Texas is putting on a show.
Mm.
So you can come and see us sing, and it's going to be awesome. And we're going to have a couple of quartets come in from out of town. So Google Acapella, Texas, and you can buy tickets right on the front. And it was great to see all you guys today. It's always a good time.
Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Brianne Barker's at Drew University Bioprof Barker on Twitter. Thanks, Brianne.
Thanks. I learned a lot.
And Angela Mingarelli is at McGill University. Thank you, Angela.
Thank you. It was a great time.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Jolene for the timestamps, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We will be back next week. Another TWiV is viral.